Welcome you to the Congressional Biomedical Research Caucus. I'm Peter Kairos, representing the Joint Steering Committee for Public Policy. We're so happy to see you on such a day that's not as hot as yesterday. You know, we had a long season this year of caucuses, all about Alzheimer's, cancer, and we hope at any time for any of you to give us any topic of great interest to you so we can provide it to our speakers, and we'd be delighted to have any suggestions. You know, in the last 4 or 5 years, we were fortunate to have the chairs of our caucus, Sherry Boulder, Mike Castle from Delaware, Lois Capps, and Rush Holt, really helped in doubling the NIH, and that was a real achievement, and all of you really helped, and it was a great achievement. But this year, as you all very well know, for the first time in nearly 40 years, as long as I can remember, when I was in the Congress, the NIH always used to get 4 or 5% every year. This year, the NIH is really flat funded. And it's really a terrible thing because it doesn't provide for economic growth, doesn't provide for competitiveness, doesn't provide for young men and women to come along and become research scientists. And furthermore, it doesn't provide for the cutting edge research that you're so used to. So we're really concerned, and we understand already that Senator Spector, yesterday, in markup, in subcommittee on labor HHS, in the Senate side, did put in $200-odd million more for the NIH. So sometime in the fall, there will be a conference between the House and the Senate, maybe after the elections from all we hear. We want you to be alert and do whatever you can to help. Now today, we're going to learn about ways to help the immune system recognize and destroy cancer cells. For the past 40 years, scientists and clinicians have tried to harness the immune system to attack cancer in order to avoid the destructive side effects of conventional therapies. And this groundbreaking work is now in clinical trials, and earlier reports show that many promising responses have been detected in many patients with late-stage melanoma as well as kidney, ovarian, and prostate disease. So we're really privileged today to have with us a world-renowned scientist in the field of tumor immunology. Dr. James Allison is chairman of the immunology program, Memorial Sloan-Kettering Cancer Center, received his BS and his PhD from the University of Texas, from where he hails in Dallas. And then he was a postdoc fellow at the Scripps Clinic and Research Foundation. Dr. Allison has a long-standing interest in the mechanisms of T-cell development and activation, and the development of novel strategies for tumor immunotherapy. He was the first person to isolate the T-cell antigen receptor protein, a feat which has been called one of the 3 most important findings in immunology in the last 20 years. So he has made many contributions to our understanding of T-cell activation, and we're looking forward to listening to him this morning. Dr. Allison.
Thank you, Peter. So let me get back to start. Yes, as Peter mentioned, I'm going to talk about harnessing the immune system. It is something that people have been trying to do for a very long time. And as many of you who read the newspapers over the years are aware, I'm sure there have been anecdotal successes, but on balance, it really hasn't been all that successful, mainly because we didn't really know what we were doing. So what I'm going to talk about today is some recent advances in basic science that were made in my lab that enabled us to learn enough about how the immune system is regulated, to learn new tricks to mobilize the immune system. Now, there have been successes, like with monoclonal antibodies, for example. I'm sure you've heard of Herceptin, used to treat breast cancer, and rituximab used to treat lymphomas. These are immunological in a sense because you raise antibodies that are specific for cancer cells. You can attach drugs to them, but that's not really what I'm talking about. What I'm talking about is actually trying to mobilize the immune system in the patient. Recently, preventative immunization has become a reality with the first vaccine to human papillomavirus, which should go a long way towards eliminating cervical cancer in this country. And that's quite an achievement. But again, that's preventative, that's prophylactic vaccination. And what I'm going to tell you about today is some things that we've learned about how we can hopefully do therapeutic vaccination to patients that already have cancer. And The general area I'm going to talk about again is regulation of immune responses. One of the things that we didn't realize until the last 5 years or so is that the immune system has a very complex set of regulatory circuits that are there to keep it from destroying normal tissues, from causing autoimmunity. And we call these checkpoints. And what we found basically is a way to block some of these checkpoints to allow the immune system to really do its thing in an unfettered manner. So just to give you a little primer about the immune system, this shows an infected cell, but it could be a cancer cell with mutated proteins in the cell. T cells are really cool because they have antigen receptors, as we showed about 20 years ago, that actually recognize, as shown here, magnified, bits of proteins that represent everything that's going on inside a cell. And these are taken up by molecules and displayed on the cell surface. And T cells have a set of receptors that can recognize those little bits of protein. And they cruise around the body, bumping into your cells all the time. And if they see something that's wrong, that T cell gets basically a signal to take off, to divide and differentiate into a cell that can actually kill, for example, virus-infected cells. So I've really been interested in T cells for that reason, because the tumor product or whatever does not have to be on the surface of the cell. It can be anywhere in the cell. And the immune system can be able to detect that. And this just shows what can happen. This is a scanning EM showing a tumor cell here, and these are T lymphocytes that are sitting on it, and they can actually punch holes in the membrane of the tumor cell and also deliver enzymes that will go in and chew up the DNA of the tumor cell, leaving it nothing but a little hollow sphere here, little filaments that make up the cell membrane. This is the T cell after killing it. So this can work in vitro, and the trick, of course, is to make this happen effectively in the person that has a tumor. Now, as I told you, the T cell, when it recognizes this little bit of protein displayed on the surface of the cell, gets a signal. But it was shown in the late 1980s, actually, by some intramural scientists at the NIH, that signal by itself is not enough to get a T cell going. You can think of this sort of as the ignition switch of a car. You know, every car's got a different one. You put the right key in there and the engine starts, but nothing happens because you've got to give it a push. So there's another molecule that has to be engaged. It's called CD28. And we showed in 1990, actually, that you have to get a second signal through this CD28 molecule at the same time that you're getting a signal through the antigen receptor to get a T cell actively going, to get it to really divide. And one thing that's interesting is that tumor cells don't express the molecules that engage CD28. So they're basically invisible to the immune system until something happens that I'll show you in a moment. But when we clone the gene encoding this molecule, again, this is all basic studies, have nothing to do with cancer when we're doing this, we found that there's another molecule called CTLA-4 that comes up after this other molecule. It's induced when a T cell starts getting activated, and its function actually is to turn the immune response off. And it sort of serves as the brake to the immune system. And this is a really important molecule. We and others knocked the gene for that out. We mutated the gene and made mice in which that pathway was defective. And they die because their immune systems destroy them. So this is the critical checkpoint in downregulating immune responses. And again, all this work was carried out in very basic studies and work funded by grants from the NIAID and from the NCI over a long period of time, without sustained funding to look at these. We discovered that molecule in 1982, this one in 1990, and this one in about 1995. So this is a long process involved in getting there that required a lot of support from the NIH. So I hope you will take what you heard earlier to heart and support further funding. In any event, one of the things we did in working this out, as I'm going to show you here, is we tagged these two molecules with fluorescent dye so that we could watch them. And what you're going to see here is what happens. This is a T cell coming in contact with the tumor cell here. And you can see this CD28 molecule moving into the structure that's called the synapse. That's the region of the cell that's giving the signal that's going to tell that T cell to start dividing. And you can see how quickly that happens. The red is just a false color image showing that the protein is concentrated. But CTLA-4, again, our basic studies show that it has a very different biology. It's located inside the cell in little vesicles in the back of the cell. And so once the cell gets started by the T cell antigen receptor signaling and CD28 signaling, then CTLA-4 molecules are mobilized from the other end of the cell. They're drug around to the front of the cell, and they start shutting the response down. And again, this is done to keep your immune system from attacking you. If you don't have that happen and limit the response, bad things will happen. So at any rate, after working out the biology of this in the basic studies, the thought occurred to us that we should be able to exploit this to really, again, mobilize the immune system. According to the scheme here, the idea is that, again, tumor cells may have these antigens on their surface that could stimulate T cells, but they can't give that second signal. So what has to happen is some tumor cells die, as happens all the time. As a tumor mass gets to a certain size, it has limited access to oxygen and food and whatever, and some of the cells just start to die. That gives an inflammatory signal, alerts cells of the immune system we call antigen-presenting cells. It's sort of a circular argument, but these are cells that are called phagocytic cells. They go out and they'll gobble up, literally, bits of the dying tumor cell, process them, and display that tumor antigen on their surface, now with these molecules that stimulate CD28. So this happens at low levels all the time, and that, making this happen better, is one of the challenges for modern tumor immunology. People are trying to develop vaccines because now we know about 100 or so of the molecules that the immune system can see on tumor cells, so you can actually think about injecting those signals themselves to prime. But I would submit to you, one of the things that we're really excited about is if any way you kill a tumor cell, by chemotherapy, as long as it's a chemotherapeutic drug that spares the immune system, by radiation, by hormone therapy in breast cancer or prostate cancer, or by blockade of angiogenesis, something that Judah Folkman's pioneered, all of those are going to kill tumor cells. All of those are going to make this process happen. But inevitably, for the reasons that I told you, you're going to induce the CTLA-4 molecule, it's going to come up and it's going to limit that response. And in some cases, it'll actually stop the T cells dead in their tracks, again, in an attempt to try to avoid autoimmunity. So we had the idea that we could, if we could block that pathway, take it out of action temporarily, for example, with an antibody that we made in the mouse, we could take the brakes off, if you will, allow this T cell receptor signal and the co-stimulatory signal provided by CD28 to be fully realized and get the T cells just to go nuts for a time. And this was attractive to us for several reasons. The first is, as you can imagine here, it doesn't matter what kind of cancer it is. This should work against all kinds of cancer cells. The second one, as I mentioned, is we don't have to think of this as some kind of specialized therapy. It may work by itself and take it by monotherapy and taking advantage of the death that's occurring at low levels of tumors all the time. But it should also synergize with other immunotherapies and more conventional therapies. And finally, to the extent that you dysregulate this pathway temporarily while the antibody's there, antibodies usually have a half-life of a couple of weeks or 3 weeks in the circulation. It'll normally go out of the system and then it all comes back into normal regulation. So we tried this in mice, block it to get unrestrained proliferation. So we tried this in mice, and we're able to do this because in mice, there are genetically identical strains of mice, and there are tumor cell lines of a large number of tissue types that have been established. And you could inject them into mice and let them start growing, and then we could inject the antibody that we made, the monoclonal antibody to CTLA-4, and sometimes we would use a vaccine as well, and then we just watch the tumor grow. And we can do these in large numbers of mice, and that are, again, all genetically identical, so we can get results that hopefully we could repeat. And this shows the first experiment we did. This is a colon carcinoma that's growing, again, on the back of a mouse. If we don't inject anything, the tumor grows and we have to euthanize the mice after a little over a month. If we block that CD28 molecule, that gas pedal, if you will, the tumor actually grows faster. It reaches a larger size faster, showing that the immune system's trying to do something, but it just can't do it. But if we block the CTLA-4 molecule by injecting antibodies in those mice, the tumor grows for a couple of weeks and then gets rejected. And those mice are permanently immune. This is one of the things that we're after in immunotherapy, because the T cells that kill the tumor stay in the body, and if a tumor comes up again, a micrometastasis anywhere in the body, the T cells will seek it out and kill it. You know, you don't have to worry about whether it's surgically accessible or whether it's a place where you can irradiate or anything like that. Anyway, those mice are permanently immune, and it's not because the antibody is doing anything to the cancer cells, because if we deplete the killer T cells, which we can do, if we remove those from the mice, that antibody doesn't do anything to the tumor. So again, it's not working on the tumor cell, it's working on the immune system. And we've done this with many different kinds of tumors and many strains of mice, and it works a lot of the time, but not all the time. This is a particularly nasty tumor. It's a melanoma called a B16 melanoma. A few hundred B16 cells will kill a mouse. It metastasizes to the lungs, just as human melanoma does, and is very poorly immunogenic. Before we did these experiments, people had been trying for 20 years to do something immunologically against this tumor, and it had never really worked. It was just so poorly immunogenic. So here, as you can see, when we injected the antibody, it did nothing to the tumor, really. But a colleague of mine, Glenn Dranoff at the Dana-Farber Cancer Center, had shown that if you put a gene called that encodes a molecule called GMCSF, it doesn't matter what that stands for, but that attracts those phagocytic cells to the region of the dying tumor. If you use that vaccine, which are tumor cells with that gene in it that are then inactivated by the radiation, that also doesn't work with this tumor. It worked with some other ones, but it didn't work with this tumor. But if we combine these two things, neither one of which did anything whatsoever to this tumor on their own, 0 plus 0 in this case equals 100%, because now all the mice are cured of this tumor. And again, they're permanently immune, and if we deplete CD8 T cells, it doesn't work. So again, we've amplified now 2 points. We've amplified the presentation of antigens to the immune system and then taken the brakes off the T cells so that they go out and lysate. I just make this a point because the FDA used to require that before you could combine two agents, you had to show that each of them had some efficacy individually. And that's really nonsense, because here's an example, a very clear example of two things which by themselves don't do anything, but if you understand how they work, you can put them together and get, incredible synergy, as I'll show you here. Anyway, if you look inside these tumors, this is a tumor just happily growing along in an untreated mouse. This is a melanoma. These green cells are a kind of T cell called helper T cells. The ones with the purple nuclei are actually suppressor cells. These, we know now, are recruited by tumor cells, and these guys actually turn off other components of the immune system by a mechanism we're studying now. But we can blow right past that with our therapy, because after we treat with CTLA-4 blockade, you can see these red cells have gone into the tumor, and those are the killer T cells. They ignore the suppressive effects of these guys. They go into the tumor and kill it. In 2 weeks, this mouse is going to be dead because his tumor is too large and has got lung mets. In 2 weeks, there's going to be nothing left on this mouse except a little bit of scar tissue where the tumor used to be. And so, in any event, we've not found any experimental tumor in mice that we can't cure by some combination of other treatment with anti-CTLA-4. And again, many of them, the antibody works by itself. Well, when again we did this basic work, I was at the University of California at Berkeley. We didn't have a medical school. We wanted to start doing clinical trials. We didn't have access to patients. So what we did was to make mice that expressed the human CTLA-4 molecule by getting a piece of DNA and splicing in the human sequence that's on the outside of it. And then we introduced that into the mice that we had knocked the gene out. So we have now mice that express the human molecule that we can actually test drugs in to be used in humans. So this shows an experiment. This is a repeat of the first experiment I showed you. Now that antibody to mouse CTLA-4 doesn't do anything, of course, because the antibody to the mouse molecule isn't there. But a mouse antibody that we made to human CTLA-4 is pretty effective, and a human anti-human CTLA-4 antibody that was made by a company called Medarex that I consult for, completely blew the tumors away then in the mice. So now we have a molecule that we know, or an antibody that works with the human molecule in these mouse clinical trials, if you will. And so the mouse data were exciting enough where Medarex then took this into primates. The antibody cross-reacts with monkey CTLA-4 and showed that it caused no harmful side effects in monkeys and actually enhanced immune responses to model antigens in monkeys. So then it went into a phase 1 clinical trial. And I'm just going to show you one patient from that. There were 12 patients with stage 4 metastatic melanoma. This is lethal disease. All these patients had failed every other therapy. This woman had failed every other therapy, including another vaccination strategy using dendritic cells pulsed with tumor antigens. This is how she presented. You can see this grapefruit-sized lung metastasis and this pleural effusion. This is just a bunch of loose tumor cells in her lungs. This is after a single injection of antibody with no side effects. 5 months later, greater than 98% of her tumor was gone. About 6 months after that, she got another injection of antibody. The rest of the tumor went away, and that was 5 years ago. And she's had no recurrence. Anyway, there were 3 patients that had partial responses out of the first 12 in that trial. There were also 3 out of 7 patients in late-stage prostate cancer who showed drops in PSA, again, after a single injection of antibody. So this got everybody pretty excited about this, and now it's actually in phase 3 trials in metastatic melanoma. It's been picked up by Bristol Myers Squibb and is being co-developed by Bristol Myers and Medarex, and is in phase 3 trials. I said we hope it's been fast-tracked by the FDA, and we hope it'll be licensed within the next year or so. But I want to show you now examples some additional trials, phase 2 trials, that show that this works against melanoma and other cancers. So one of the trials, one of the early combination trials, was actually done by Steve Rosenberg and his colleagues at the surgery branch of the National Cancer Institute here in Bethesda. This shows one of the patients in the first dozen he treated. This was a young man who had 31 lung metastases. You can see some of them here. And so he was treated with the antibody plus a peptide vaccine. After 3 cycles, all of his lung mets were gone. He also had a brain metastasis that you can see here. And after 3 cycles of treatment, that was also gone. That was almost 4 years ago now, and he's had no recurrence since then. That's the good news. The bad news is that this fellow actually rejected his pituitary for reasons that we don't understand. So he's on maintenance therapy to deal with that, and that reverses the effects of it. But he's fine. His cancer is completely gone. And again, in the first 12 patients in this trial, there were 3 objective responders like that. So Steve's group has gone on to treat, as you can see here, over 100 melanoma patients, up to 200 now, and 61 renal cancer patients. And the data is basically the same for each. About 1 in 6 or so patients have an objective response. That is, and by objective, what I mean is what's called the RECIST classification. That means 51% reduction in the total body load of tumor. And many of them had complete remissions. That is, tumors were undetectable after the therapy. And the exciting thing, because I'm sure you read about stuff in the papers all the time, as I do, where you do a trial, and if it extends life by 60 days or 90 days, it's considered a success. These remissions last for years in these patients. They're very durable. But there are side effects given this way. I should point out that these patients got the antibody at either 3-week or 1-month intervals repeatedly until they develop side effects, because that's the way oncologists like to do this. That's not the way an immunologist would like to do it, but that's the way an oncologist does it. So there is a price to pay, but all of these adverse immune reactions that the patients have, again, are reversible with cessation of treatment and with symptomatic support. So recently, this is a trial that's actually still going on in the Netherlands. It's supported by Medarex and Bristol Myers Squibb and a company called Cell Genesis. It uses that same GMCSF vaccine that I showed you that we used in our mouse experiments, given at 2-week intervals, coupled with antibody every second dose. And this sort of trial is a dose escalation. They start with a sub-effective level of antibody and then are gradually increasing it. This is at the 3rd dose level, where you can see all 3 of the patients. Again, you do 3 patients and then you wait and see what happens. And if nothing bad happens, you go to the next dose. But all 3 of these patients, 3 of 3, were partial responders, or were objective responders in the sense they had greater than 50% drops in their PSA. And these are patients with hormone-refractory metastatic prostate cancer. So these were men with a life expectancy of 6 months or so at the time that they were treated. You can see this fellow's PSA basically went to 0. And so there was a bone scan done on this fellow. As you know, prostate cancer tends to metastasize to the bones. This shows that that rib is really loaded with tumor. There's a spot in that rib which completely went away following the therapy. And then these large mets in the vertebrate and the pelvis and the top of the femur all are greatly reduced in size. And that shadowing may just be healing of this. So this fellow has had, again, an objective response. Actually, at the next highest dose level, there were 2 of the patients that had bone marrow clearing, and one of them that cleared a metastasis that was in a lymph node on the background. So anyway, the total there at the 2 highest levels was 5 out of 6 objective responders receiving this treatment. But again, there were some side effects. These fellows developed a partial hypophysitis, it's called, and temporarily at least have to be on support. But again, as I mentioned, the antibody is given every month, and that's sort of the old way of doing things, giving the drug until it makes you sick, and then hopefully the doctors try to cure the side effects and then hope the tumor goes away. But you don't have to do that. Again, my colleague Glenn Dranoff, who pioneered the GMCSF vaccine approach, likes to take a different sort of approach where he immunizes. What he actually does is to harvest tumors from the patients and then genetically engineers the patient's own tumors to express this GMCSF molecule, again irradiates them and puts them back into the patient. And he's been doing this in ovarian cancer and melanoma and lung cancer for a time, and it hasn't really worked very well by itself. It's shown by this. This is an 81-year-old woman with disseminated ovarian cancer. CA-125 is a serum protein that's made by ovarian cancer cells. It's much like PSA. It's just used as an index of total body tumor load. Her CA-125 levels were climbing. Glenn made a vaccine from her tumor cells. As I mentioned, immunized her over a 2-month period and it did nothing. She was basically becoming moribund from the tumor in her abdomen. So a month after the last vaccination, Glenn gave her a single dose of antibody and just waited. And she continued to worsen for a couple of months, and then her CA-125 dropped from almost 5,000 to less than 100. And this was accompanied, you can see here, these solid tumors in her abdomen and this large metastasis in her liver. Those went completely away, and this one went down very much in size. So she had an objective response both biochemically and by scanning, and she also had symptomatic relief because, believe it or not, she felt so good she went out and had hip replacement surgery here. And so in a couple of months her CA-125 went back up and she was sick again. She got an antibody here, but this was after several months, but no vaccine again, and her CA-125 again fell. At this point she felt so good she had cataract surgery, believe it or not. And anyway, you can see about every 4 or 5 months she gets another dose of the antibody, and she's been a lot, this started in March of '03. She's still doing fine. She's 84 now. And so the worst case, and she had no side effects, I should point out, except for a mild skin rash that resolved on her shoulders that resolved without any treatment. So if we can reproduce this is what I see us doing in the future, is instead of just trying to bomb the tumor and nuke it by frequent administration of antibody, making people sick, if we could just spread that out and gradually mobilize the immune system, kills a bunch of tumor cells, the antibody goes away, the CTLA-4 pathway comes back into check, but then when you give the antibody, you've got more cells the next time, you've got more cells to work on. So you can see she's responding faster every time she gets the drug, so it's working better every time she gets it, not worse, as usually happens with most cancer therapies. So anyway, that's what I want to show you, how this basic science that we've been doing actually led us to have enough insight into the regulation of these processes where we could take this checkpoint that's critical and that obviously limits immune responses to cancer and block it temporarily. And so where we're at now is trying to figure out as a monotherapy what distinguishes the people that respond from the people that don't, and we don't have a clue. Except that there are other checkpoints. My lab just cloned a gene about 2 years ago for a molecule that's actually put up on the cell surface of many cancer cells that blocks their killing by cytotoxic T cells. And so now we're exploring whether that would be a good checkpoint. Trying to discover the best vaccine to use in combination with this. That GVAX approach works pretty well. And what conventional therapies could be used. In the mouse, we've shown that it synergizes with hormone therapy and with radiation and with some chemos. And finally, my real dream, and the way I think this field is going, is to combine it with the new targeted drugs that you've heard about, the kinase inhibitors, Gleevec and Tarceva, which is used for lung cancer. These drugs have received a lot of press, and they're amazingly effective because they will kill almost all the tumor cells. But the key is they'll kill almost all the tumor cells, and inevitably patients have recurrences with these drugs because the tumor mutates the kinase that those drugs inhibit. And so when the tumor comes back, that drug doesn't work anymore. And we think that if we combine our tricks and take advantage of the priming of the immune system that happens when you have that first bolus killing of tumor cells by these targeted drugs, if we can mobilize the immune system at that point, we could get immunity which will take out the rest of the cells and prevent that recurrence that occurs because they mutate their kinase. So that's where we're going with this in the future. And I just won't mention everybody, but I want to thank my research team here at Memorial that contributed to this work. And with that, I'll close and be glad to take any questions that you have. So thank you for your patience.
Thank you very much, Dr. Allison. Now we'll take any questions that you might have. And so I'll ask the very first one. What about geriatric patients? You mentioned a woman 84 years old. Does the immune system stay effective as people grow?
That was a real surprise, actually, because it is the thymus that produces T cells, involutes, when you're about 40 years old, and it's usually thought that your immune system's gone downhill. And it was a real surprise that she responded so well. But since then, there have been 2 elderly gentlemen at Memorial that were treated with this, and their thymus, there's some evidence their thymuses actually regenerate under the influence of this drug. So it may reverse we don't know if that's for sure yet. Their thymus has certainly increased in size. But we think that this may just sort of restore proliferative capacity to the...
Because Jim, I was thinking a lot of older men get prostate, and
Right.
You also mentioned prostate being treated, but the immune system will still be there for them. All right, where does CTLA-4 come from? Where does this antibody come from? How do you produce
Well,
it?
we made the original one against the mouse molecule because we'd cloned the gene, so we just expressed it actually in a hamster cell line and then injected that into hamsters, and they made an antibody response to it because it was foreign, and then we cloned the cells that were making the antibody, and we can produce it forever. What Medarex did to develop the drug that's used in people is they have mice that they've replaced the human, sorry, they have mice where they've replaced the mouse antibody genes with human. And so they did the same thing. Then they immunized those mice with a recombinant protein that encodes the CTLA-4 molecule. The mouse make an immune response, except those antibodies are human. And then they clone the genes for those from the B cells, from those guys, and then put them in cell lines, and they just crank out the antibody in tissue culture. And fermenters basically the same way you make beer. So.
Yes, please.
Hi.
Want to wait for the microphone, please, ma'am.
Thank you. I didn't read the slide completely, but the slide about Dr. Rosenberg's patient said something about him developing hepatitis. What was that about?
There was one patient that developed an immune inflammation in the liver. And this fellow had been on alpha interferon therapy for years, which itself causes some liver damage. So we think that was a manifestation. So the usual, there are side effects. I mean, particularly in the patients that are getting the antibody at intervals less than a month. In the melanoma and renal carcinoma patients, it was usually colitis. It was a very bad gut reaction, which could be treated with corticosteroids. The second most frequent side effect was this immune attack on the pituitary gland for reasons we don't understand. In the prostate trials, the pituitary toxicity is the predominant one, again, for reasons that we don't understand. But in Glenn Dranoff's patients and other patients that have received the antibody at bigger intervals, there really are no side effects other than mild skin rashes.
Is this research funded by the NIH? And also,
Absolutely.
Are you involved with clinical trials with the NIH?
Yes, so absolutely. All of the basic research that we've done over the years on all these topics was funded by grants from the NIH. I'm proud to say I have a grant that's in its 30th year now. I'm one of those rare
30
people
years?
Yeah, that I've been funded. So close to 30, maybe it's 20 now. Anyway, I had it back in Texas before I ever went to. So anyway, it's absolutely without the NIH support, we wouldn't have been able to do any of it. We don't, many of the trials are funded indirectly by the NIH. What Medarex and Bristol Myers did is provide the antibody to the NCI, and people can apply to get it for trials. They write a proposal, and if it's peer reviewed and it meets the usual criteria, then the NCI actually gives the antibody to the investigators. So there are about 70 trials going on now. All over the world, actually, but all over the country for sure, many of which are funded by the NCI.
Yes, sir.
Are you doing any research to compare the genetic profiles of responders and non-responders?
Yes, that's just started, actually. Medarex has got a big effort there.
Explain that, would you?
Well, there are 1 in 6 people respond, and there's possible that there are genetic differences in the CTLA-4 molecule itself. There have been some polymorphisms, that is, changes in the gene sequence. That have been shown in populations. And so one of the things is to see if that correlates with whether the antibody works or other genetics things. There are about 100 genes that are being tested in responders and non-responders to see whether that's going to enable us to, before treating, define or identify those people who've got a good chance of responding.
Yes, sir.
Yes, I was wondering if you could look into your crystal ball and tell us, in your estimation, when will immunotherapy start to displace current treatment modalities, surgery and chemo?
Yeah. Well, I'm not sure it'll ever displace them. I think it'll make them work better. This particular drug, I hopefully it's been fast-tracked, as I mentioned, by the FDA. So hopefully within a couple of years it'll be available for melanoma. Once that happens, people are going to start using it for everything. So I think I would hope that within 5 years or so. I mean, we're starting the animal models now, the animal experiments with Gleevec and Tarceva to see if that's going to work. So we'll have to see, but I think within 5 years we'll see this really go mainstream.
Yes, Ms.
I
Please.
have 2 questions. One is, have you had difficulty recruiting people into the clinical trials? Is that extending the length of time to do your research? And then I had a question about your mouse models and whether they were generally males or if you had both females and males.
Okay. The first part, you know, I'm not directly involved in the clinical trials. I should have made that clear. One reason is that I hold intellectual property on this, which has been licensed from the University of California, and so I've got to stay out of the trials. I give advice, but I don't, quite the contrary. I think through word of mouth now, I get calls from clinicians all over the country saying, what about this? You know, my patient heard about this and they want it. You know, there's an incredible communication between cancer patients on the internet, and the word's spreading, and they're, you know, I know of plenty of doctors, particularly in Boston, at Dana-Farber, that get calls, people begging to get on this, so that hasn't been a problem. And yes, in mice, we tested both males and females.
Jim, you mentioned 4 cancers: ovarian, prostate, colon, and renal. Now, what is the significance of what you're telling us today? Will this new antibody really give relief for tumors that occur there that other people couldn't treat in any other way?
I certainly hope so. I mean, everybody that does immunotherapy over the years has traditionally started with melanoma because it's an inflammatory tumor and there's good evidence of immune responses. But that's not been true of ovarian cancer. That's not been true of prostate, and yet this works. So I think it's got great promise for virtually any kind of cancer, frankly, except those. We are worried about the immune system attacking tissue-specific things, you know, so probably wouldn't want to do this right away until we have more experience of cancers of tissues that are absolutely indispensable to survival. You know, heart, muscle, you know, brain, stuff like that.
And what about the time frame? Will this drug be available widely for use very soon?
I hope, as I said, I think within depending on how this big trial accrues, within 2 to 3 years, because it's been fast-tracked by the FDA based on the initial data and the lack of serious toxicity, unmanageable toxicity.
Pam?
Are these same mechanisms that you've been working on at the basic level for so many years applicable to figuring out what's going on with autoimmune diseases?
Absolutely.
That's on the flip side of what you're doing.
Absolutely. So while we're in a whole another line of experimentation, we've been trying to develop ways to specifically engage the CTLA-4 molecule when we're giving an antigen. We've been doing this in a mouse model of multiple sclerosis and in a mouse model of diabetes, of autoimmune diabetes. And we're not there yet, but absolutely, it could be, if we can figure out how to do it. In theory, it should be doable.
Well, Dr. Allison, thank you very, very much for this privilege. We want to thank the Joint Steering Committee for Public Policy for sponsoring this lunch. And let me tell you about our next caucus. It will be held on July the 26th on a Wednesday. The subject is going to be systems biology and new science from a great Harvard professor. Thank you all very much.